Thank you, Stephanie. Sorry for that pause as I'm getting it together. Okay, for our next award, we have the Raymond L. Lindeman Award, which recognizes a young scientist each year for leading an outstanding peer-reviewed English language paper in the aquatic sciences. The 2022 Lindeman Award is being given to Dr. Mohammad Monir Moniruzzaman in recognition for his 2020 Nature paper, Widespread Endogenization of Giant Viruses Shapes Genomes of Green Algae, describing the integration of viral elements from giant DNA viruses into the genomes of green algae, providing evidence of their importance in protozoan evolution and response to environmental change. Dr. Moniruzzaman is currently an assistant professor at the Rosenstiel School of Marine and Atmospheric Science, University of Miami. This paper significantly changed the field's understanding of how green algae evolved by demonstrating the incorporation of viral DNA contributing many genes to these protozoan genomes. The discovery described herein, or in this paper, constitutes a mechanism of evolution previously unappreciated in these organisms. This work provides very large advances that have opened new and exciting avenues of research in aquatic microbiology. Please join me in welcoming, there you go, Mohammed to the stage.
Thank you everyone.
And I don't know where.
I'll just.
Okay, great.
How do I cancel that? Hold on. Escape? Okay. Yeah, give me 1 second. Just this. I have this open.
Yeah. Oops.
Okay, so that goes right. Yeah. Now, yes. Yeah, thank you. Hello, everyone. Thank you very much for being here today, and it's a great honor to receive the award. I was just looking at the boards here, and I saw my PhD supervisor was an awardee in 2021. Made me really emotional, and it puts a lot of pressure on me. I started my position in University of Miami in January, not too long ago, just 1 semester. So I guess I was very surprised to receive this award. I did not expect it at all. And it really also put things in perspective and also kind of helped me to realize that there's a lot of things to do for me. There's a lot of responsibilities ahead. Currently, I'm a professor, assistant professor at the Rosenstiel School of Marine and Atmospheric Science at University of Miami. And the paper that received this award, it was published in Nature in 2020, and this is part of my postdoc work with Frank Aylward in Virginia Tech. So I'll be talking about giant viruses, green algae, and how these viruses can shape the genome evolution of this algae. So lots of things to digest, and I'll try to make it simple and straightforward. So please forgive me if you think that I'm being technically too, relaxed or something. Yeah. So I call this gentle giants because what happens is that when these viruses get in the phenomenon that I'm going to describe, there's no war, there's no killing going on. The viruses are actually living peacefully within the genomes of green algae. So before I go start, I need to introduce the viruses, actually, because what is a giant virus, right? We all know virus, and we all know what they do and what they're doing right now, right, to the world. But what is a giant virus? What is, how many of you recognize this, the inspiration of this picture? That's the Beatles walk, right? So the picture basically gives you an idea of, I'll try to use this and see if I can. Can I? Okay, no worries. So, this is just to give you an idea of how big these viruses are. The entities are drawn to roughly to the scale of size. So you have E. coli, it's a very famous bacteria, we all know about them. And then we have all these giant viruses, Tupan virus, Pythovirus, Pandora virus, Mimi virus. They are all, and you can see that they're pretty big. I mean, they're almost as big as bacteria, some of them are. So that's why they're known as giant viruses. Now, the natural question is, do they infect bacteria? No. They infect eukaryotes, mostly single-celled eukaryotes that we call protists. And some of them can be as big as 1.5 micron in diameter, and the genomes can be as big as some bacteria, like 2.5 megabase pairs. That's the largest recorded genome size for these viruses. And what's interesting about the genomes of these viruses is that they have lots of genes. Some of them encode more than 1,000 genes, individual genes. And a lot of these genes have very interesting evolutionary history. For example, we know that some of the giant virus genes have origins in bacteria. But I told you that they don't infect bacteria, right? So why do they have bacterial genes in them, for example? That's a current question that we're trying to answer. It's ongoing research, and a lot of people are working on these things. Many of these giant virus genes encode cellular functions. For example, photosynthesis, nutrient metabolism, nutrient uptake, energy metabolism, for example. And I would not go into details about the functions of these genes today, but one of my major research questions that I want to answer in my lab is actually to characterize some of the functions of these viral genes and how they actually contribute to the host virus infection dynamics. Now, so as I said, the giant viruses, they're briefly called, technically they are called nucleocytoplasmic large DNA viruses. In short, they are called NCLDVs, and I will try to use that term throughout the talk. That will refer to the giant viruses, basically. So what do they do? They infect many eukaryotic lineages, primarily microbial eukaryotes that can include primary producers, that can be grazers in the marine and freshwater ecosystems. And some of them also infect animals like fish and other crustacean pathogens. Some examples, you can see the pictures here. Micromonas is a green algae, single-celled green algae. Cafeteria, the second picture, is a heterotrophic grazer, protist grazer. Then we have choanoflagellates. Lots of this brown algae or kelp that we know in the ocean, they are also infected by viruses in the same family. I have seen a lot of talks and a lot of posters in this conference about algal blooms, and you'll be surprised to know that a lot of the algal blooms actually have associated giant viruses in them. As an example, this is the Emiliana huxleyi bloom. That's the coccolithoper bloom that you can actually see from the space. It's so big in terms of area that it covers. And during the collapse of the bloom, the viral activity increases dramatically, and that virus is a giant virus. And we have seen this kind of viruses in other blooms. For example, brown tide blooms in the Long Island area. And finally, we have also seen association of these viruses, NCLDVs, with coral. There's a lot of research going on, and to characterize the role of viruses in coral bleaching and stress response, and among the viruses, a prominent component of coral holobiont is this NCLDVs, or giant viruses. So moving forward, I'm going to give a quick primer of the classification of these viruses a little bit, otherwise it'll be tricky to actually follow the talk. So these viruses share a common ancestor. So they all evolved from an ancestral virus. Most of these viruses share some hallmark genes, that is genes present across all the viruses in this group. We'll call them viral hallmark genes. There are like 7 to 8 genes like that we can use to construct phylogeny of these viruses, to understand their classification, their diversity. So this is a figure from one of my work in my postdoc that we published in 2020. And this is basically, in this work, I recovered about 500 novel NCLDV genomes from environmental metagenomic datasets. And I know it's a complex figure. I won't go too details into it. But this basically is the phylogenetic tree of these viruses. Inside this circle, you see the tree, and each branch represents one virus, right? I would say that the takeaway from this figure is that the majority of these viruses belong to two different groups. One is the green group, that is the Phycodenaviridae, that's on the top right. And then the whole purple group, that's the Mimiviridae, that's another group. And from this tree, it's pretty obvious that those are the major groups of giant viruses that are present in the environment. And the size range, this bar plot around the circle that represents the size of individual viruses. From this, you can see that there's a distinct difference of size between Phycodenaviridae and Mimiviridae, right? And they can go as high as 2.5 megabase. And in the mimivirus group, there's a lot of viruses that are up to 500 kb or longer than that in genome size. So that's an idea of diversity of these viruses and what they do. So now move on to the next project, the project that that I did in my postdoc and for which the award I received. So this is it's like I don't know, it's like looking back, it was totally just because I was curious about this phenomena, and some luck was involved, actually, for this project. So the figure here you see, it's basically just telling you that it's a blast search figure. Basically, when you have these genes, individual genes, you can go to NCBI and look at the sequence database. They have thousands to millions of sequences from individual organisms and viruses in NCBI. So in my PhD, I used to just, make phylogenetic tree of these viruses. So for that, I had to use this database frequently. Whenever I'm looking at individual genes for these viruses, I always see hits to eukaryotic genomes. I expect hits to viral genomes, which is obviously fine, but then I also see hits to eukaryotic genomes. And that made me really annoyed. Why do I see all these hits to eukaryotes? These are viral genes. Where are they in the eukaryote? What's the reason? So initially I thought that, well, maybe one or two viral genes got integrated in the eukaryotic genomes by horizontal gene transfer, which is common across the tree of life. Then also it made me think that if viral hallmark genes are present in these genomes, what about the other genes? What about the other genes in these viruses? So, and I always asked myself, I was trying to think that can I determine the extent of endogenization? Can I determine how many viral genes are integrated in this host genomes? Is there a way to do it? Hallmark genes are pretty obvious. They are viral genes, we all know about them. But as I said, that these viruses have hundreds and hundreds of genes. And we don't know how to determine that if they're integrated in these genomes. So my postdoc supervisor, I have to give a shout out to him, Frank. Frank said, okay, well, Monir, if you want to work on this, you can work on this. I have projects for you in my mind, but I will allow you to do whatever you want to do. So I was literally just given total freedom in pursuing my research question. So to talk about the project, why we chose green algae? Why did we look at the green algal genomes? Well, the obvious reason, I found the hits initially in some of the green algae. So that motivated me to look deeper into these genomes. And at that time, there was a lot of publicly available genomes for these green algal lineages. The reason is that a lot of these genomes are sequenced because there's increased interest in biofuel production with this different algae. So that allowed me to actually mine these publicly available genomes for these viral signatures. Methods are complicated, takes time, but in brief, what I did is that I used a combination of criteria to define the viral sequences within the algal genomes. Some of the criteria includes that, okay, I looked at distinct nucleotide composition that is different from the host-specific sequences. So sequences that have very different nucleotide composition that would signify or indicate that these are probably sequences that's coming from a foreign source. It's not the eukaryotic sequence. I looked for the viral hallmark genes in this kind of sequences, and also checked if these genes had best hits to known NCLDV genes or virus genes. We integrated all this criteria in a bioinformatic tool that we call viral recall. That comes from Total Recall, if you recognize. And we used that tool to automate the search. Now, long story short, again, complicated figure, but basically this circle plot on the left, that is representing all the viral sequences that we defined within the genome of one green algae, that's Tetrabina socialis. All this together almost represent 2 megabase pair genes, genome sequence within the Tetrabina. That's a green algae. We found lots of duplications in these sequences. We found lots of introns. Now, introns are specific to eukaryotic sequences. The fact that the viral sequences have introns means that these viruses invaded the green algal genomes, and the introns, afterwards, they invaded the viral sequences because they're mobile elements. So basically, it tells you that these sequences are actually part of the host genome now. But at some point, they were individual independent viruses. Now they become endogenized in the host. So basically, this is the endogenous giant virus, which we call GIVs, or giant endogenous viral elements. Now that's Tetrabina socialis. If we move forward, we actually found 2 different virus signatures in Tetrabina socialis. So there was like 2 individual genomes of giant viruses in one algae. And we also found the same thing in Chlamydomonas eustigma. That's the 2 circles on the right. Those represent the viral genomes integrated in Chlamydomonas eustigma. That's another green algae. So not 1, but 2 distinct giant viruses invaded these 2 green algae. And in terms of the contribution, like how dramatic they are, that in Tetrabina, about 10% of all the genes of Tetrabina are actually coming from giant viruses. That's a lot of viral genes that define the genome complement of an algae. So briefly, I just want to mention that we eventually found 18 individual GIVs that were represented in 12 green algal genomes. So some algal genomes had more than 1 GIVs. 14 of them has some kind of phylogenetic affiliation. 10 comes from the Mimiviridae group, and 4 comes from the Phycodinaviridae group, the groups I talked about in the beginning of the talk. And for 4 of these GIVs, we could not determine where they're coming from because they don't have any hallmark genes in them. So now, just to wrap up, what's the implications? Why do we care, and why is it interesting? Initially, I mentioned that a lot of these NCLDVs have genes that come from different sources, including bacteria. Now, when these genes get into these algal genomes, that means they're working, like they're functioning as a massive conduit of horizontal gene transfer. Lots of genes are being transferred from external sources to these algal genomes. And that can happen, that can have like multiple implications. Now, we are at the very beginning of this research. We just discovered this, and we don't know yet exactly what these genes are doing in these genomes. But we can speculate. We can, form some working hypothesis. So we think that some of the genes brought in by these GIVs can actually, give the algae opportunity to co-opt some novel functions, like some functions that the algae doesn't have, but the virus has. So the algae can co-opt maybe a nutrient transporter that the virus is bringing in, and then algae can use that to actually, start more efficiently transporting nutrients, for example. Of course, these viral signatures will shape the pan-genome of this algae. That is, like within a species, we will see some strains having these viruses, and some strains will not have these viruses, and that will shape the pan-genome dynamics of the overall species. And that kind of variation can actually lead to phenotypic differences within the species and also adaptation to different environmental pressures. And one of the favorite hypotheses that I have is that some of these GIVs might actually help this algae to resist subsequent viral attack. And there are evidence for that in not algae, but other organisms, that integrated viruses can actually help the organism to prevent further attack by viruses. But we have not, we're kind of pursuing this with a model system right now, but I can't share the results at this moment because I don't have it. So to wrap up, I think that this kind of endogenization of giant viruses in green algae is very interesting, and it's also likely very common, and represents a very exciting new research field. So with that, I just want to thank my lab. I know I'm running out of time here. Frank has been incredibly helpful, and he's a new PI, and I enjoyed my 3 years in Virginia Tech working with him. We're like buddies and sharing ideas and stuff. And all the students, Carolina, Alina, they also contribute to this project. Thank you very much.